So chapter 13.3 talks about how meiosis actually happens, all the steps that take place and everything. And the important thing is that the entire goal of meiosis is to change the cell from diploid to haploid. So when we're done, we only want the cell to have one copy of each of the chromosomes. So for human cells, that would be one copy of each of the 23 chromosomes, which is different than the rest of your diploid cells, which all have two copies of each of the 23 chromosomes. So in order to divide the cell at all, we need to replicate the chromosomes. So this is still going to happen, just like during mitosis. All of the chromosomes are replicated. And then rather than the cell just dividing one time like mitosis, it divides twice, which is how we get 4 daughter cells. So we start off with 1, we divide it in half once, then we have 2, we divide each of those in half again, then we have 4 daughter cells. And each of these daughter cells are haploid. So there's, like I said, 2 cell divisions. So the first one is called meiosis 1, the second one is called meiosis 2, and each of these has the same steps as mitosis. So there's prophase, prometaphase, metaphase, anaphase, and telophase, just like in cytokinesis, just like in mitosis, and it happens twice. So we call them prophase 1, prometaphase 1, prometaphase 2 when we get to the second one, etc. And so these are the important things that happen in them. In meiosis 1, the homologous pairs of chromosomes line up. So for example, your chromosome 1 from your mom and your chromosome 1 from your dad are going to line up and separate. And then, so after meiosis 1, our cells are haploid. So that happens during meiosis 1. And then during meiosis 2, we pull apart the sister chromatids, just like we did in mitosis. And the result is 4 daughter cells, different than mitosis, which gives you 2. So you have 4 daughter cells, and each of these are haploid, and they each have 1 chromosome of, for example, human cells each have 23 single chromosomes. Okay, this is a really good picture that your textbook has. It's on page 253. And this shows what happens to the chromosomes during meiosis. So this is in the original parent cell. There's a pair of homologous chromosomes. This could be chromosome 23, or it could be chromosome 1, or it could be chromosome 12, whatever chromosome it is. You have 1 copy from your mom and 1 copy from your dad. Okay? So this is a diploid cell. There's 2 copies of each of the chromosomes. Then we replicate them during S phase. Okay? Just DNA replication. We'll talk about all the logistics of that later, but the chromosomes are replicated. So now we have 2 sister chromatids attached in 1 chromosome. So it's the same identical information, just duplicate copies of it. So now we have these are homologous chromosomes. Dad, mom. They are exactly the same. They contain exactly the same information. So this one right here is going to be the gene for your eye color. Right here is going to be the gene for your hemoglobin. Same exact thing on this one. Eye color, hemoglobin. Exactly the same. Then we get to meiosis 1. During meiosis 1, the homologous chromosomes are separated. So the chromosome 1 from your dad goes into one cell. Chromosome 1 from your mom goes into a different daughter cell. So that is what actually happens during meiosis, is we separate the homologous chromosomes. So now each of these cells only has genetic information from 1 parent, meaning it is haploid. Okay, we are diploid up here. Now we're haploid after meiosis 1. Then we go into meiosis 2, and we separate the sister chromatids just like we did during mitosis. So here's the identical copies of the genetic information from your dad. They get pulled apart during anaphase. Over here from your mom during anaphase. So now we have haploid cells with unreplicated chromosomes. So you have 1 copy of each gene in each of the daughter cells during meiosis. So before meiosis 1, there's interphase, just like there is in mitosis. And the chromosomes are duplicated and we get sister chromatids, just like we saw here. So this is interphase, so we replicate the chromosomes. The sister chromatids are genetically identical, just like in mitosis. They're still attached at a centromere. And we replicate the centrosome, which is where we make the mitotic spindle. So that's what happens during interphase. And then meiosis 1 has 4 stages. So right here we're not talking about prometaphase. That's okay. So we have prophase 1, metaphase 1, anaphase 1, and telophase 1, cytokinesis as well. And let's look at each of these pictures. Okay, so here's prophase 1. The homologous chromosomes are lined up, or paired up, different than the sister chromatids, okay? So they are forming what's called a tetrad. So there's 4 chromosomes together. Look at this right here. So here this is, let's say this is chromosome 3. You have your chromosome 3 from your mom, chromosome 3 from your dad, and they are attached to each other. The homologous pairs are together. And since there's 4 of them, because they're replicated, there's 4 sister chromatids right here, it's called a tetrad. Is that what your book's calling it? A tetrad. So, and then the same thing is going to happen. Nuclear envelope breaks down, and the mitotic spindle attaches to the tetrads instead of just the chromosomes. Then the tetrads during metaphase 1 get pulled into the metaphase plate. Then during anaphase 1, the homologous pairs get pulled apart. This is what's different than mitosis. In mitosis, the sister chromatids get pulled apart. In meiosis 1, the homologous pairs get pulled apart. Then the cell finishes dividing. I don't know what that says. I lost it. I don't know what that says right there. I'll tell you later. And so we divide the cell, and then we have now 2 haploid daughter cells with replicated chromosomes. Okay, they each only have this. Let's say this cell n number is 3. 1, 2, 3. That's all they have. They do not have duplicates of their chromosomes, but the single chromosomes are replicated. So we still have 2 sister chromatids in this chromosome. So prophase 1 takes up 90% of the time required for meiosis, and this is where the chromosomes begin to condense, and the process by which the homologous chromosomes pair up is called synapsis. And these are lined up exactly. So let's say, here's my 2 homologous pairs. Let's say eye color is right here. It's going to be in exactly the same spot right here. They line up exactly and correctly, and that's really important, and we'll talk about why that is later. And the reason that that's important is because something called crossing over happens, and that comes in the next section. And these are called a tetrad. Tetra means 4, so there's 4 sister chromatids attached, and sometimes these the homologous chromosomes cross over each other, which is what is called a chiasmata, but we're going to talk about that in the next section. Then metaphase 1, like I said, all the homologous pairs line up along the metaphase plate. The spindle fibers from each end of the cell attach to either side, just like they did in metaphase during mitosis. And during anaphase, the homologous pairs separate. 1 chromosome moves into each pole, or to each side of the cell, and the sister chromatids are still attached, so that's really important. They're still shaped like this. They haven't divided yet. Then telophase 1 in cytokinesis. Each cell is haploid, but they are still replicated, and cytokinesis happens at the same time, pinches the cell in half. We have 2 haploid daughter cells. Same thing as mitosis. In animal cells, we have a cleavage furrow. In plant cells, cell plate forms. Then the cell goes into like a brief pause phase, kind of like a second interphase, but we don't replicate the chromosomes again because we don't want to do that. The purpose here is to divide them, so we don't want to do that again. So that's what's different. Then we have meiosis 2 again, and this is more similar to mitosis in the way that things happen, and we again have prophase 2, metaphase 2, anaphase 2, and telophase 2. And this is what it looks like. Now the sister chromatids line up along the metaphase plate. You can see it's happening twice because we had 2 daughter cells from meiosis 1. So they line up along the metaphase plate. Each sister chromatid is attached to a centrosome via spindle fibers at either ends of the cell. During anaphase, they get pulled apart. And now, finally, after telophase 2 and cytokinesis, we have 4 daughter cells. They are haploid because they all only have 1 copy of the genetic information, and they are all different, and that's really important. Don't worry, we're going to talk about crossing over in the next section, so anything that's about that we'll come back to. You can read all this. It's the same thing as mitosis. That's what it said. It's basically exactly the same as mitosis, except it happens twice because we already had 1 division, and we're pulling, and each of the daughter cells is haploid, and they're not identical. In mitosis, they are diploid and identical. Okay, so this is where it gets tricky, is comparing mitosis and meiosis. Your book does a really, really good job of this. This is on page 257. So during mitosis, we keep the chromosome number exactly the same because you need to have all genetic information inside of all of your cells. So you don't want half of your cells to like have 92 chromosomes, the other half to have 23, some of them to have 46, right? We want them all to be the same. So that's why it's really important during mitosis. But in meiosis, we divide the chromosome number in half. We go from 2N, 46, to N, 23. We go from diploid to haploid, and they're all different from each other. And that's really important that they're different from each other. And that, again, comes in the next section. This is a great figure, so make sure you are paying attention to this. Basically, during mitosis, sister chromatids line up on the metaphase plate. And then in meiosis, during metaphase 1, homologous pairs line up on the metaphase plate. And then during mitosis, or metaphase 2, sister chromatids line up on the metaphase plate. Make sure you read this. There it is again. Okay, and there's 3 things that are unique to meiosis, and all of them happen in meiosis 1, because remember meiosis 2 is really similar to mitosis. So during prophase 1, the homologous chromosomes are pulled together, and remember they form a tetrad. Now, these 2 homologous pairs are going to kind of switch genetic information. So they're shaped like X's, right? What they do is they like cross over like this, and then they switch what's on the top. And this can happen up to 3 times, I think, on each chromosome. So they're just like swapping genetic information all over the place. And that's called crossing over. And that happens during prophase 1. And that's what leads to genetically identical, or genetically distinct daughter cells. So they're all different. So that is the first thing, is crossing over. And it's called synapsis when they cross over. So at the metaphase plate, we're pairing homologous pairs, not sister chromatids. That's different than mitosis. And in anaphase 1, the homologous pairs separate, not the sister chromatids. So those are all things that we already talked about. So the reason that the sister chromatids stay together during meiosis 1, when they don't during mitosis or during meiosis 2, is because there's a bunch of proteins that attach them together. They're called cohesions, but that's important to know that there's a reason why they don't separate right there. And that is all of the steps of meiosis.